Chlorella is a genus of single-cell green algae belonging to the phylum Chlorophyta. It is spherical in shape, about 2 to 10 meters in diameter, and is without flagella. Chlorella contains the green photosynthetic pigments chlorophyll-a and B in its chloroplast. Through photosynthesis, it multiplies rapidly, requiring only carbon dioxide, water, sunlight, and a small amount of minerals to reproduce. The name Chlorella is taken from the Greek comma chloros, meaning green, and the Latin diminutive suffix -ella, meaning small. German biochemist and cell physiologist Otto Heinrich Warburg, awarded with the Nobel Prize in Physiology or Medicine in 1931 for his research on cell respiration, also studied photosynthesis and chlorella. In 1961, Melvin Calvin of the University of California received the Nobel Prize in Chemistry for his research on the pathways of carbon dioxide assimilation in plants using chlorella. Many people believe chlorella can serve as a potential source of food and energy because its photosynthetic efficiency can, in theory, reach 8%, comparable with other highly efficient crops such as sugarcane. Chlorella as a food source: it is a potential food source because it is high in protein and other essential nutrients. When dried, it is about 45% protein, 20% fat, 20% carbohydrate, 5% fiber, and 10% minerals and vitamins. Mass production methods are now being used to cultivate it in large artificial circular ponds. It is also abundant in calories and vitamins. When first harvested, chlorella was suggested as an inexpensive protein supplement to the human diet. Advocates sometimes focus on other supposed health benefits of the algae, such as claims of weight control, cancer prevention, and immune system support. According to the American Cancer Society, available scientific studies do not support its effectiveness for preventing or treating cancer or any other disease in humans. Under certain growing conditions, chlorella yields oils that are high in polyunsaturated fats. Chlorella minutissima has yielded EPA at 39.9% of total lipids. History: following global fears of an uncontrollable human population boom during the late 1940s and the early 1950s, chlorella was seen as a new and promising primary food source and as a possible solution to the then current world hunger crisis. Many people during this time thought hunger would be an overwhelming problem and saw chlorella as a way to end this crisis by providing large amounts of high-quality food for a relatively low cost. Many institutions began to research the algae, including the Carnegie Institution, the Rockefeller Foundation, the NIH, UC Berkeley, the Atomic Energy Commission, and Stanford University. Following World War II, many Europeans were starving, and many Malthusians attributed this not only to the war, but also to the inability of the world to produce enough food to support the increasing population. According to a 1946 FAO report, the world would need to produce 25 to 35% more food in 1960 than in 1939 to keep up with the increasing population, while health improvements would require 90 to 100% increase. Because meat was costly and energy intensive to produce, protein shortages were also an issue. Increasing cultivated area alone would go only so far in providing adequate nutrition to the population. The USDA calculated that, to feed the U.S. population by 1975, it would have to add 200 million acres, 800,000 square kilometers, of land, but only 45 million were available. One way to combat national food shortages was to increase the land available for farmers, yet the American frontier and farmland had long since been extinguished in trade for expansion and urban life. hopes rested solely on new agricultural techniques and technologies. Because of these circumstances, an alternative solution was needed. To cope with the upcoming post-war population boom in the United States and elsewhere, researchers decided to tap into the unexploited sea resources. Initial testing by the Stanford Research Institute showed chlorella, when growing in warm, sunny, shallow conditions, could convert 20% of solar energy into a plant that, when dried, contains 50% protein. In addition, chlorella contains fat and vitamins. The plant's photosynthetic efficiency allows it to yield more protein per unit area than any other plant. One scientist predicted 10,000 tons of protein a year could be produced with just 20 workers staffing a 1,000-acre, 4-square-kilometer, chlorella farm. The pilot research performed at Stanford and elsewhere led to immense press from journalists and newspapers, yet did not lead to large-scale algae production. Chlorella seemed like a viable option because of the technological advances in agriculture at the time and the widespread acclaim it got from experts and scientists who studied it. Algae researchers had even hoped to add a neutralized chlorella powder to conventional food products, as a way to fortify them with vitamins and minerals. When the preliminary laboratory results were published, the scientific community at first backed the possibilities of chlorella. Science Newsletter praised the optimistic results in an article entitled "Algae to Feed the Starving." John Burlew, the editor of the Carnegie Institution of Washington book Algal Culture from Laboratory to Pilot Plant, stated, "The algae culture may fill a very real need, which Science Newsletter turned into future populations of the world will be kept from starving by the production of improved or educated algae related to the green scum on ponds." The cover of the magazine also featured Arthur D. Little's Cambridge laboratory, which was a supposed future food factory. A few years later, the magazine published an article entitled "Tomorrow's Dinner," which stated, "There is no doubt in the mind of scientists that the farms of the future will actually be factories." Science Digest also reported, common pond scum would soon become the world's most important agricultural crop. However, in the decades since those claims were made, algae have never been cultivated on nearly that scale. Current status: since the growing world food problem of the 1940s was solved by better crop efficiency and other advances in traditional agriculture, chlorella has not seen the kind of public and scientific interest that it had in the 1940s. Chlorella has only a niche market for companies promoting it as a dietary supplement. Production difficulties: the experimental research had been carried out in laboratories, rather than in the field, and scientists discovered that chlorella would be much more difficult to produce than previously thought. To be practical, the algae grown would have to be placed either in artificial light or in shade to produce at its maximum photosynthetic efficiency. Also, for the chlorella to be as productive as the world would require, it would have to be grown in carbonated water, which would have added millions to the production cost. A sophisticated process, and additional cost, was required to harvest the crop, and, for chlorella to be a viable food source, its cell walls would have to be pulverized. The plant could reach its nutritional potential only in highly modified artificial situations. Another problem was developing sufficiently palatable food products from chlorella. Although the production of chlorella looked promising and involved creative technology, it has not to date been cultivated on the scale some had predicted. It has not been sold on the scale of spirulina, soybean products, or whole grains. Costs have remained high, and chlorella has for the most part been sold as a health food, for cosmetics, or as animal feed. After a decade of experimentation, studies showed that, Following exposure to sunlight, chlorella captured just 2.5% of the solar energy, not much better than conventional crops. Chlorella, too, was found by scientists in the 1960s to be impossible for humans and other animals to digest in its natural state due to the tough cell walls encapsulating the nutrients, which presented further problems for its use in American food production. Use in carbon dioxide reduction and oxygen production. See also, carbon sequestration. In 1965, the Russian SILS experiment BIOS-3 determined that 8 square meters of exposed chlorella could remove carbon dioxide and replace oxygen within the sealed environment for a single human. The algae were grown in vats underneath artificial light. Alternative medicine. Chlorella is consumed as a health supplement primarily in the United States and Canada and as a food supplement in Japan. Chlorella has a number of purported health effects, including an ability to treat cancer. However, according to the American Cancer Society, available scientific studies do not support its effectiveness for preventing or treating cancer or any other disease in humans. Health concerns. A 2002 study showed that chlorella cell walls contain lipopolysaccharides, endotoxins found in gram-negative bacteria that affect the immune system and may cause inflammation. However, more recent studies have found that the lipopolysaccharides in organisms other than gram-negative bacteria, for example in cyanobacteria, are considerably different from the lipopolysaccharides in gram-negative bacteria. A 2010 study found that chlorella contains a peptide, known as Chlorella-11, that actually inhibits the inflammation caused by lipopolysaccharides from gram-negative bacteria. Aquaria. Chlorella can create green and opaque water problems in aquaria. Chlorella can grow due to high nitrate and phosphate levels or direct sunlight. Decreasing phosphate and nitrate by partial water change and moving the aquarium to shade can help alleviate the problem.